morning. My name is Nancy Padilla-Coreano, and I'm an assistant professor at the University of Florida, where I've been for the last 2 years leading my research lab. And my mission is to transform our knowledge of how we navigate our social worlds using this with a mouse model. And today I'll be telling you about the work I've done for the past 5 years, some of this work as a postdoc in the lab of KTI, and some of this work in my research lab. So I'll be telling you about the neural circuits underlying social competence. So what is social competence? We're capable of optimizing our social interactions based on the available information. Like when you're with friends, you act a certain way that you don't act when you're at work in an important conversation or having an important conversation in a parenting scenario. So as a social species, we're very much able to optimize our social behavior based on the available information, who's there, and the context. And this is crucial for our survival and thriving. And we're not the only social species that's able to show social competency. Many other species show the ability to adjust their behavior based on who they're interacting with and the context. And specifically, social dominance hierarchies provide a very important information of like the relative ranks of the animals and how they should act with each other. So we've known about dominance hierarchies for more than 100 years when it was first observed as the pecking order in chickens, but we still have a very poor understanding of how the brain enables dominance behavior and how it enables us to know, keep track of these social ranks. And why is this important? This is important because many neuropsychiatric disorders disrupt the ability of individuals to navigate social groups. And these disruptions are very detrimental for physical and mental health because we are social animals and we really need those social interactions. And I believe that if we focus on understanding the mechanisms that guide social behaviors in the healthy brain, this will help us understand those social disruptions and dysfunctions that occur in many neuropsychiatric disorders. Now, back to the idea of social dominance. What do I mean by that? How does it manifest? Imagine you're at a work party and there's one slice of pizza left and you and your boss both reach for it at the same time. Now, if you're not your boss, you're probably going to retract your hand automatically. So your brain does that almost automatically. Like it recognizes who they are, the context, and that because they have more power over you, perhaps you should let them have the pizza. So social dominance is defined as basically that, consistently winning at points of social conflict when the motivational priorities of the two individuals are in competition. So it's basically winning in a social competition setting. Now, how do we study how the brain enables this social dominance behavior in a social competition setting in the lab? How do we do that? We actually use the mouse as a social species that shows social dominance and competitive behavior, both in the wild and in the laboratory. They're a great model to study this question. And more specifically, even in laboratory settings, they form very stable social hierarchies that we can measure and study in a lab setting. Now, when it comes to where in the brain could there be happening important computations to control this behavior, the prefrontal cortex is key because it integrates information and guides social decision making, and there's evidence for this across species. And more specifically, the medial subdivision of the prefrontal cortex, what we call the mPFC, is linked to social dominance across species as well. And here's 2 examples. One study in humans shows that mPFC activity correlates with social rank learning in an fMRI setting. And one study in mice shows that if you optogenetically manipulate and drive activity in the mPFC, that's sufficient to change social dominance, to increase it in a competitive and increase the competitive success of the individuals with that stimulation. So this really links the mPFC to social dominance and winning in a social competition setting. But how is this happening? How is the prefrontal cortex encoding the social rank information and flexibly adjusting the competitive behavior? And this is the question that my work gets to. And throughout this talk, I'll be talking about 3 different lines of research that help us understand this big picture question better. The first one the first part of the talk, I'm going to show you some evidence that shows that mPFC neurons at the population level encode competitive success and social rank. Next, I'll be showing you some evidence that a top-down circuit, specifically the prefrontal cortex projection to the hypothalamus, to the lateral hypothalamus, controls competitive success and increases competitive success. And finally, I'll be showing you some very recent and preliminary work from my lab where we're considering a large network of regions that are projecting to the prefrontal cortex and important outputs of the prefrontal cortex to understand how social competition could be encoded at the network level. Now, behaviorally, how do we measure social rank and how do we study social competition in the lab? We first use a very well-established method of getting the social ranks of males that are caged together, the tube test. So in groups of animals that are caged together, they naturally form a social hierarchy, and we just need to be able to read out that hierarchy. So one method of doing that is with the tube test, where you put two mice, and the tube is so narrow that they cannot cross if there's two of them. So then the one that pushes out the competitor out is actually dominant based on many other metrics, like their agonistic behavior, their urine marking, and this is being published multiple times and validated. So we first got the social ranks of stable hierarchies, and then we individually train each mouse to associate a tone with a reward. And after each mouse of a given group really understood that tone reward association, and then we brought them back to this context where they were getting the tone, and this time in the presence of a cage mate of a known social rank. So then this created a context of social competition because for every tone, there's only one small drop of reward. So both of the animals cannot get it. And what we see is, as expected, that animals with higher ranks have a higher probability of getting their reward. But importantly, this is not a binary thing. The dominant animal does not get every single reward, which enables us to look at the dominant brain when it's losing and the subordinate brain when it's winning, and really dissociate winning versus losing versus dominant and subordinate. And overall, this task is really like enabling this pizza scenario, but in a controlled manner and repeat it so we can get that trial-based information. And this is a video of what it looks like. So here we see the mice are wearing these wireless devices, and this is how we actually record from the prefrontal cortex. And this is the port that delivers the reward, and this light indicated that the tone turned on. So this is the trial that this animal got the reward, the one in the yellow, and then it will be a losing trial for the one in the blue. And these wireless devices are basically a miniature electrophysiology rig on the head of the animal that enables us to record populations of mPFC individual neurons. This device amplifies, digitalizes, and stores the data. And at the end of our experiment, we just extract the data from the head of the animals and isolate individual neurons just like any other extracellular recording. And this is just example cells that have been well isolated. So using this approach, we ask, what are the mPFC neurons doing during social competition? What are they responding to? And for that, we consider the main task-relevant events that are happening during this social competition. And the main event is really the tone, because the tone is signaling the availability of the reward. So we're looking at tone onset, what we're considering the trial onset. However, it's not the same to hear a tone when you're about to win or when you're about to lose that trial. So we separate the responses based on the outcome of the trial. We call these win versus lose trials. Another important thing that's happening is that the animals enter the port to pick up the potential reward. And not only is the subject entering the port, which we call self entries, but we also have the competitor entering the port, which we call other entries. So we've got the self entries and the other entries, and we looked at the neural activity in both of these situations. And these port entries can happen, there's initiated by the animal, they can happen whenever the animal wants. So sometimes the animals enter the port during the tone when there's reward available, but actually during competition, they also enter the port when the tone is not playing in the intertrial intervals as well, because this is a strategic behavior that enables them to potentially be at the port when the tone starts. So we also looked at neural responses of these reward-seeking strategic behavior. And I'm going to show you some highlights of some of the things that we found for these events. One of them is that even though the same number of neurons respond to winning in dominant and subordinate animals, when we look at how the neurons are responding, they're responding slightly differently, with subordinate animals having stronger mPFC excitation during winning. And that's the lighter color that you see here. Now, in the case of reward-seeking behavior, those self-entries and the other entries, like the competitor entering the port, what we see is that there's actually more cells in the dominant animals that encode this reward-seeking behavior for both the self and the competitor's behavior. So these are pie charts showing the number of neurons that are significantly changing firing rate to these events. And you can see that there's more neurons for dominants versus subordinates in both of these cases. And even though dominants have these larger ensembles that are encoding for reward-seeking behavior, when we consider how the responses are happening, actually subordinates have stronger responses to the competitor's action. So these are the responses in the subordinates when their competitor enters the port when the tone is playing and when the tone is not playing. And in both of these cases, we could see that there's larger magnitudes in the response of the firing rate of the subordinate neurons. So this means that overall, mPFC neurons are showing important information regarding the social competition, both the tone onset and the behavior of the animals for seeking out these rewards. And importantly, it's also encoding the behavior of the competitor when they're seeking out rewards as well. And interestingly, even at the level of individual neurons, we see that social rank modulates this response. Now, we wanted to next understand how could this be affecting a population level encoding of social rank and competitive success. So for studying the population dynamics in the mPFC, we did dimensionality reduction, so that way we can explore the population dynamics in both subordinate and dominant animals in the same subspace. So for this, we basically combine every single neuron we recorded into a pseudo population. So this means like across every single animal, no matter what their social rank is. We do a dimensionality reduction with all of the activity of these neurons such that we can visualize them in the same subspace. And we did this for the times surrounding the tone onset. And we separated the data by winning or losing. And we have 5 seconds of time before the tone onset and then 5 seconds after the tone onset. So in this plot, you see in this dimensionality reduced space doing principal component analysis, you could see that the data is separated by the outcome of the competition. These are the trajectories in blue of when the animals win and the trajectories in red when the animals lose. And I'm going to walk you through each of them. So this is the trajectory for when the dominant animals win, when the dominant animals lose, when the subordinate animals win, and this is the trajectory of the population when the subordinates lose. So first, we can draw a line separating winning versus losing data. And we could draw another line that's almost orthogonal to that line to separate the data based on relative rank. And very interestingly as well, the neural trajectories are separate even before the onset of the tone. So the triangle indicates the onset of the tone, and the circle is 5 seconds before the tone. And these trajectories of winning and losing are already separate. And that's the case as well for subordinates, suggesting that perhaps the prefrontal cortex has some predictive information of competitive success for future competitive success. However, this data comes from average firing rate across the whole session and across trials. So in order to really know if there's any predictability or encoding of mPFC for future competitive success and also for the social rank of the animals, we had to employ a supervised machine learning approach where we can take a single time point in a single trial and ask if we can decode whether the animal is going to win or lose, or whether the animal is dominant or subordinate. And this is the performance of both of those decoders separately. The one on the left is decoding whether the animal is winning or losing, and 0 is the onset of the tone. So you can see even 30 seconds before the onset of the tone, we can predict better than chance and better than shuffle data if the animal is going to win or lose. And we can very stably and very well predict the relative rank of the animal throughout the trial. And we believe that this is this higher performance for decoding relative rank is because the relative rank of the animal is stable throughout the session, but whether the animal is winning or losing actually is dynamic across trials. But importantly, this suggests that mPFC at the population level does have predictive information of the future competitive success of the animal and the social rank of the animal. Next, I'm going to switch gears and show you some circuit data, identifying the prefrontal hypothalamic circuit as one that is important for competitive success. Given that mPFC has this competitive success and social rank information, we wanted to understand how could this be changing behavior. And two potential subcortical pathways that we wanted to explore are the lateral hypothalamus and the basolateral amygdala, because there's evidence that the lateral hypothalamus regulates social behaviors, but also reward and avoidance behaviors. And there's evidence that in non-human primates, BLA firing rate is predictive of social rank, of the social rank of the faces that the monkeys see of animals they know. So these are two potential targets of how mPFC information could be sent subcortically to change behavior. And in order to consider these two subpopulations, we did something called phototagging to identify the specific neurons of the mPFC that project to those downstream regions and record from the enduring behavior. So this was done separately in animals for the lateral hypothalamus versus the basolateral amygdala. And in this cartoon, I'm showing you the lateral hypothalamus example. So we first inject a retrograde virus that carries Cre into the downstream brain region. And then the mPFC axons that are terminating in that region are going to pick up that virus. And then we're going to put a secondary virus in the mPFC with an excitatory opsin that is Cre dependent. So only the neurons of the mPFC that project to the lateral hypothalamus or the BLA in the other animals are going to express our excitatory opsin. And the idea is that during, while we're recording in vivo, we're going to shine pulses of light and identify which neurons respond to the light with a monosynaptic photolatency, with the principle that neurons that have the opsin are going to respond very, very quickly to the pulses of light, but neurons that don't have the opsin are not going to be light responsive or going to respond much later with polysynaptic latencies. And so we record from animals during the competition, and then at the end of the recording, we shine the light. That way we're not affecting the behavior with our stimulation. And then this is just an example neuron that was phototagged. So these little rows are basically when we shine the pulses of 5 milliseconds of the light, and you can see how regularly this neuron responds to the light with a couple of milliseconds of latency. So we recorded from mPFC-LH neurons and mPFC-BLA neurons and asked what is happening during the competition. And these are specifically trials where the animal is getting the reward, so winning trials. And we see that mPFC-LH neurons are being excited by winning trials compared to mPFC-BLA neurons. And that's not the case when the animals are alone and are just receiving their reward outside of the context of competition. mPFC-LH neurons do not respond to the just receiving the reward in an alone context. So we were interested in understanding if perhaps these mPFC-LH projecting neurons modulate competitive behavior. So for that, we turned to optogenetics to stimulate this pathway of the mPFC to the LH in the animals that were of lower relative rank during the competition to see if that would affect their competitive success and their behavior during the competition. And when we quantified this, we saw that actually the animals that received this mPFC-LH stimulation were able to win more trials compared to when the light was off. And we did a lot of control experiments to rule out the possibility that this was due to changes in the hunger or changes in their food-seeking behavior or even their reward-seeking behavior when they were alone. So we really think that this is enhancing their competitive abilities. So just to conclude, what I've shown you so far, mPFC encodes social rank and future competitive success at the population level. And even at the individual cell level, we see that the mPFC is encoding the important task-relevant events that are happening during social competition, and social rank is already changing how those neurons are responding. And we identify the mPFC-LH projectors as a potential group of neurons that encodes winning. And the mPFC-LH pathway modulates dominance behavior. And now I'm going to switch gears to completely new data collected in my lab in the last few months. So this is very much preliminary and completely unpublished direction, where we want to understand where is this prefrontal information coming from and where else could it be going? More specifically, how does the network work together during the social competition? We know that the mPFC receives inputs and sends outputs to other regions that are important for social cognition and this social competition context. For example, we know that the medial dorsal thalamus input to the mPFC shows plasticity during social competition, specifically during winning, when the animals win in competition. We know that the ventral hippocampus projection to the mPFC is important for social memory, and perhaps social memory could be playing a role in these behaviors, given that there's a hierarchy established. And we know that BLA shows social rank information and also social valence coding. But the mPFC also sends multiple outputs, not just to the lateral hypothalamus, but it's interconnected with some of these same brain regions that send inputs. So to assess how everything might be working as a network, we're simultaneously recording mPFC single cell activity and then local field potentials in all of these distributed network to understand how this prefrontal subcortical network encodes social competition. So the idea is that by recording simultaneously local field potential and single unit activity, we can assess both these neural oscillations that occur at different frequencies in the local field potential and how they relate to spiking activity in the mPFC. For example, this is a neuron that is spiking in the peaks of the oscillation that's filtered in red. So that would be a phase-locked neuron. And we can also assess functional connectivity across different brain regions. For example, in this trace example of hippocampus and prefrontal cortex, this is a high synchrony moment where the phases of the two local field potentials are more in phase. And then this is a low synchrony moment. And we can measure that with coherence. And we can also look at directionality of these signals. Like in this example trace, the hippocampus is leading the prefrontal cortex. And to compare the network in a baseline condition versus this competitive state, we recorded in the same animals when they're in competition with a cage mate and when they're receiving the reward alone. And when we looked at our network state, one really interesting thing that we saw was that synchrony increased during winning, specifically for mPFC and the LH, and mPFC and the basolateral amygdala. So that's the blue line here. And the synchrony increase specifically in the higher frequencies. So beta and gamma frequencies show the increase. And the dark blue line is the winning trials, and the lighter blue is the alone rewarded trials. And you can see that the synchrony is not just due to getting the reward, but it's higher when they're winning rather than just receiving passively the reward. But this A lone rewarded condition doesn't control for the social state of the animals that we have during the social competition. So to control for that, we need a separate control where the same animals are recorded when they're in competition with the cage mate, and also when they're together, both have their own reward ports and separated by a divider for in which they can smell and interact with each other and see each other. So this is a both rewarded condition. But so it's a social non-competitive state that we can use for comparison. And so when we considered this control of both animals getting a reward, this non-competitive social state, winning was still showed higher synchrony for mPFC-LH and mPFC-BLA for the higher frequencies of beta and gamma bands relative to this control. Now, when it came to losing and what's happening in the network, one really interesting thing that we have identified so far is that the whole network is synchronizing with the ventral hippocampus, specifically in the theta frequencies. So here you can see the coherence plot for every brain region and the ventral hippocampus. And you can see how in the red line, which is the losing trials, we can see these very strong theta coherence that is higher to winning in the alone condition, specifically in the theta frequencies. Now, when it comes to the directionality of these signals of which brain region is influencing which, we measure that using grandeur causality. And I created these little summaries to show the directionality differences between winning and losing trials. So when the animals are winning relative to the losing trials, we see more mPFC in directionality influencing the subcortical regions, specifically mPFC to BLA, mPFC to LH, and mPFC to the MD. And interestingly, we're also seeing that the lateral hypothalamus seems to be a hub that influences the MD as well. Now, in these losing trials, relative to winning, we see that the ventral hippocampus has a stronger influence in the whole network. So perhaps this losing data is coming from the ventral hippocampus, and we don't know if it's associated to like perhaps a negative emotional state, but that's an ongoing hypothesis. And we're doing a lot of analysis right now in the lab to understand what changes between winning and losing states in the network to identify which of these circuits perhaps is a flip between these win and lose trials. And interestingly, when we consider the mPFC individual cells spiking relative to all of these oscillations, we find that losing induced data from these subcortical regions is very synchronized to mPFC spiking. So this shows the number of neurons that are phase locked to the different oscillations. So these are theta and then gamma. And you can see that for losing trials, we have more neurons being phase locked than for winning or when the animal is alone. So with that, we can add to our conclusions that this preliminary data suggests that losing engages theta oscillations throughout the network, and these oscillations may come from the ventral hippocampus, while winning engages a more top-down control of the network where the prefrontal cortex is leading other brain regions, and possibly oscillations of beta and gamma might play a more important role. But this is all like preliminary and ongoing project of my lab. And with that, I want to thank my lab members and the co-authors of the work I showed at the beginning and my collaborators in funding.